Hey guys, welcome back to Computational Biology with Python. In this video, we're going to continue to work with our multiple sequence alignment theme, and we'll be doing some substitutions, and we might get into some of the cluster W stuff, depending on how verbose I am and how much I talk. But let's jump right in. Okay, so we're continuing on here from our last alignment of our coat proteins. So if you're starting anew, like just rejoining, remember to reload your packages. We're going to import seq, and then from bio.seq record, import seq record, and then from bio align, import multiple seq alignment. Alright, so that's all of our required packages. We're going to say alignment equals, not a menu, multiple seq alignment, and I'm going to cheat and have you copy copy this. Okay, so pause it here, write this down. Invalid syntax, seq record. Why is that invalid? And seq record is seq seq1 equals multiple seq alignment. Is it an indentation? Whoa. I don't know what this is. Alright, there we go. I guess let's get rid of these. I don't know if it's an indentation problem. Let's try this. There we go. I guess it was an indentation problem. Python's very anal about that. So here you go. These are tabbed over twice. So just hit tab twice and that'll spread it right. So print alignment. Here's our alignment. So now substitutions equals alignment substitutions. No period at the end. There we go. And then print substitutions. There we go. So how this works is this is calculated by taking all pairs of rows in the alignment and then counting the number of times that two letters are aligned to each other. So here it counts all rows and it says when we have an A and another A, so you can see A and another A, that's 2. And okay, I said, alright, so T to T, there's 12 of them. So we got T to T is 1, T to T is 2, T to T is 3, T to T is 4, T to T is 5, T to T is 6. Then we do the same over here. But you can't count them backwards, right? Like each pair doesn't matter if it's reciprocal or not, right? So we don't overcount. T to T is, so we had 6 for the first one, and it's the same for this because there's 4, right? So T to T, 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 T to T. It's kind of like please excuse my dear Aunt Sally, PEDMAS, right? For like doing orders of operations. Where's another one? All right, so A to T is 1. and a to t is 1. So these are split evenly across this diagonal axis, right? So we should have 2 total a to t transformations. So we have a to t, and that's it. So we put 1 down here and we put 1 up here. So that's how our substitution matrix is calculated, right? It took me a minute to, like, when I was first learning, to, like, really get what's going on. I feel like I'm uncentered here. Okay, here we go. All right, so let's go n equals substitutions. Select. And we're going to select ATCG. And then, let's run that. And we're going to print so now you can see we've added a 4th substitution letter, right? Because we said that the substitutions we're interested in are A, T, G, and C. So previously we just had A and ACT for our substitution matrix, but now we're adding the 4th letter. But as you can see, since there's no, in our sequences, there's no G's, that it initially just left it off. Because it's like no G's, no point in reporting G's. We can force it to report G's, but we just get zeros because there's none in the sequence. Okay. You could also, with this, you could change, so I'm just copying and paste, you can change the order of the letters. So we'll do C and T here, and then we'll print them. So if you're like really anal about how you want things ordered, you can do ACTG as opposed to ATCG. Depends on the person. Not going to judge. Not important to me, but it's doable. Okay, let's move on. Bio align applications. All right, so let's dir directory bio align applications. So you can see that there are these algorithms out there for aligning sequences that are both pairwise alignment and multiple sequence alignments. A caveat is that these applications are generally slow, and you generally wouldn't want to write such an algorithm in Python for pairwise alignment. Biopython contains a pairwise aligner, and you can use Biopython to invoke a command line tool on your behalf, but you won't want to do a ton of alignments like on your local computer with Python. Let's see, what else? So all of these command line wrappers that we're going to talk about in this lecture, or this series of lecture videos, follow the same style. So you create a command line object, specify the options, so the input, the file name, and the output file name, and then you invoke this command line via a Python operating system, right? So like Ubuntu, or that's what we're using right now, is a Linux operating system. Let's see. Now, actually, so we're not going to explore these command line tools. We're just going to list them. We're not going to go back into command line stuff. So, but if you're interested in doing things in the command line, so remember the command line is a terminal, like this guy, that's going to take forever to, oh, wow, they load up really fast today. So if you want, you can do alignments in the terminal like this. You can do cluster W, dialign, MSA probs, command line, etc. These are all different packages, but you could do them in Python just as easy, but it's possible. I just want to put it out there, it was possible. I should have read ahead in my notes instead of saying, no, we're going to do this. But okay. Let's see, what are we at? 6 minutes only. Man, I'm going fast. All right, let's do some Clustl W. So Clustl W is a popular command line tool for MSA, multiple sequence alignments. And there's also a graphical user interface called ClustLX. The BioAlign applications module in Biopython has a wrapper for this alignment tool, so we can use it in Python. I'm going to ignore my own notes there. All right, let's go. From BioAlign applications, import Clustl W command line. Okay. Just to show you the help file. Remember, this is the help file. It's kind of a shorter help file. But if you need help, good place to go. I'm going to opt to delete that. So let's start with from BioAlign applications import Clustl W command line. And let's look at the help file real quick. Clustl W command line. Remember, this is if you get stuck on anything. We have these nice help files that will guide you through. I delete it because it takes up a lot of space in my portfolio that we're making. Alright, so let's try this. Alright, so first thing we have to do, let's go to Biopython GitHub and let's do docs. I'll post the link to this too. And we're going to use this Opuntia file here. I'm going to save it, and then I'm going to copy this and paste it so you can see it. Put a hashtag in front so it doesn't mess up my code. So that's where we're getting our file from. Then we're going to upload it, this ALN file. Then we're going to do from bio import align IO. We're going to do align equals align io.read Opuntia ALN. And we're going to say do it with a Clustl alignment. Opuntia ALN. Oh, it's probably .txt, isn't it? Let's rename this. Yeah, let's get rid of this txt. Alright, now let's try running it. Alright, so we got that. Now we're going to print our alignment. And here's our alignment. So if we open this, you can see here is our file. This is a multiple sequence alignment. So we've read it in and we've printed it. That's all we've done. So we just said align IO read and use the Clustl W. It's a Clustl W format to do that. But we can also make a tree from this. So from bio import philo. Oh, let's run that one. Tree equals philo dot read opuntia.dmd. We might have to download this. Newick. And we're going to download this file because I don't think I have how to convert it. So let me post this one too. Examples opuncha.dnd. A little tiny file. Save as. I will paste this. So we'll probably get into how to convert these, but just for this example here, we're going to remember to upload your file. We're just going to use the pre-aligned one. Rename, because it's going to have .txt in the end. Okay, now it should run. So then we can do philo draw ASCII tree. So that means ASCII means use like text, right? So here you go. So here's our tree. So you can see like if we were to open this again, this one, you can see we have these different individuals, and it's a tree file, so it just tells you distances between evolutionary distances between sequences. It doesn't give you anything in terms of alphabet, right? So once we get into the phylogeny stuff, we'll go over this a little bit more, but of how these values are created. But what you can do is you can plot these, and as you can see which sequences. You don't have annotations like if this was like, okay, human, chimpanzee, gorilla, macaque, spider monkey, or something like that. So we're like most closely related to chimps, and then to gorillas, and then to these other species. But yeah, so you can, for phylogeny, really easy to do in Python in terms of using these NUIC files. And if you took the my R class, we did a big long section on these NUIC files and drawing phylogenies as well. And I think it looks a little bit better because it's not ASCII related. So a lot of statistics and end product stuff like this, in my opinion, you'd be better off using R for. You can do it in Python if you don't want to put the time in to learn R and Python. But R just makes prettier stuff. Like, I know there's gonna be Python snobs out there like blah, 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 but like R makes prettier figures, and so this is kind of like really basic. Mid, is that what the kids say? This is very mid. Okay, so we'll stop here. This video was a bit of a train wreck for me to make. So, all right, thanks for watching again. We'll continue on. We're going to get into, we're part of my big issue. I'm not going to get into it. In the next one, we're going to use an alignment tool called Muscle. It's kind of been an update. When I was an undergrad, Clustable W was all the rage, but it looks like Muscle is now all the rage. So we're going to use Muscle for multiple sequence alignments. We'll do some subsection of muscle files, etc. So if you're watching on YouTube, please hit like, please hit subscribe. I greatly appreciate it. And I will catch you in the next video.